Okay, I think we'll get started. So, hi everyone. I'm Nandika. I'm a PhD student at IIC Bangalore, and I'll be taking the live interaction session today. So, this is the interaction regarding the NPTEL course Experimental Biotechnology, and where we'll be talking about the content covered in week two, which was basically the basics of electrophoresis. I hope everyone is able to hear me. Please let me know if you have any issues with that. Um, Again, this is a session to solve any doubts that you have and as well to demonstrate some problems and how to solve them. So if you have any doubts regarding the content, you can always ask me. You can type it in the chat or unmute yourselves and ask me. Feel free to do that. Okay. I'll just start maybe with a quick recap of what we learned this week. Okay. Okay. I hope everyone's able to see the screen. Please let me know if you can't see the screen. But yeah, we'll talk about what we learned this week, which was the basics of electrophoresis. So electrophoresis is basically the migration of a charged particle in the presence of an electric field, right? So to do electrophoresis of any molecule, it must have a charge on it. And within our experimental biology, there are a number of biomolecules which we are able to use for this purpose. That means it must have some ionizable side group. We already know that amino acids have one amino group and one acid group. Both of these are ionizable. So different pH, the amino acid will have different charges. Again, some amino acids have an ionizable side group. So that can also take on a different charge, right, a different pH. So similarly, peptides and proteins are also charged particles, right? Similarly, nucleotides and nucleic acids have a phosphate group, which will convey a negative charge to the molecule, right? So the molecule will have an overall negative charge. And how the molecule will behave in the electric field will depend on the overall net charge on the molecule, right? So I have a peptide which is made up of many amino acids. So the net charge will be the sum of all the charges which are present on the different amino acids. And that will determine how the peptide will behave in an electric field. Okay? Mainly what we are studying here is something called electrophoretic mobility, which is how it moves when we apply a voltage. So we have two electrodes and we are applying a voltage across that electrode, right? Because of this, we are generating a potential gradient. We'll call this gradient E. And this is equal to the applied voltage divided by the distance between the electrodes. In this potential gradient, any charged molecule will experience a force, okay, which is equal to the gradient multiplied by charge on the molecule. Okay? So this is the driving force that will cause the molecule to move in the electric field. So if it's negatively charged, it will move towards the positively charged side. If it's positively charged, it will move towards the negatively charged side. But it's not like it's just going to move smoothly. There will be some resistance to its motion. Okay, this is due to friction. Okay, we'll call this broadly some frictional coefficient F. Okay, and this is dependent on the properties of the molecule. So that can be the size, it can be the shape of the molecule. If it is trying to move through some medium, then what is the pore size of the medium? Is the buffer having some viscosity that will oppose this motion, etc. Okay, so there are many factors which can cause resistance, which means that even if molecules have the same charge, they will not migrate in the same way in the electric field. Okay, I have two molecules of the same charge, both have -1 charge, but one is small and one is big. The big one will face more resistance when it is trying to move. Okay, so it will not be able to move as fast in the electric field. Okay, we can describe this as the velocity of the molecule. Okay, which is basically the driving force divided by the friction. And the term that you will hear more commonly is electrophoretic mobility. Here in most textbooks, it's mu. I think in the lectures, sir has used the notation v, but in most textbooks, it's mu, which is v, the velocity divided by the potential field gradient. Okay, so this is just the basic mathematical equation which underlies what is electrophoretic mobility. Okay, The most basic way to do electrophoresis is to just have a buffer and you put two electrodes in it, right? You're not doing anything fancy. I just have a buffer. I have my mixture in it, and I'll add two electrodes. And that is called moving boundary electrophoresis, right? So there is no supporting medium. It's carried out in the solution itself. Okay. So molecules in a buffer are separated under the influence of the applied field. So this whole U-shaped tube is filled with buffer in which I have different molecules. Okay. Here there are some positively charged molecules, some relatively neutral molecules, and some negatively charged molecules. I have two electrodes which are immersed in the buffer, and according to the charge, these molecules are moving towards opposite electrodes. Okay. But you can see that this will be a very low resolution, right? Because we're just separating it based on is it positive or negatively charged. Okay, there is not much resistance to the flow of these molecules. So if I continue doing this, eventually what will happen is all the positive molecules will come on this side, all the negative molecules will come on this side. So I'm not really getting an idea of the different properties of the molecule, right? I'm just getting an idea of what is the charge. So for that, to increase the resolution of this technique, we use something called zone electrophoresis, which basically means there is some supporting medium between the two electrodes. Okay, and that can be either paper, it can be cellulose acetate, or in most cases, what we use in the lab is a gel of some sort. Okay, so gel electrophoresis we can do either in a horizontal manner, where here you can see the gel. Okay, the two electrodes are here immersed in the buffer, so there's one continuous buffer, and my gel is sitting over here, and my samples will run in this gel. The other option is something called vertical electrophoresis, where there is some buffer up here, there is some buffer down here. Okay, the two electrodes are here and here at the top and bottom, and the sample will run in a vertical direction through the gel. If you have spent any time in the lab, you will be familiar with this. This is generally how we run protein gels, and this is generally how we run agarose gels for nucleotides. So we just spent some time on what are the most commonly used gels. So one is polyacrylamide gels. Okay, if you've heard of something called SDS page, so page refers to polyacrylamide gel electrophoresis, right? This is formed by acrylamide monomer, okay, in which you've added a small amount of something called bisacrylamide, this NN-methylene bisacrylamide, okay. And what this does basically is the acrylamide polymerizes and the bisacrylamide forms a cross link between these monomeric links, okay. So basically get a 3D structure. To form this polymer, requires free radicals, for which we have to generate this free radical in some way, right? It has to be generated fresh. So we use something called ammonium persulfate and TMED. TMED is something that catalyzes the decomposition of the persulfate ion. So this is a persulfate ion. This is a free electron from TMED, which makes these two sulfate ions, or rather the sulfate ion and the sulfate free radical. Okay. This free radical is highly reactive, okay, and basically reacts with a monomeric acrylamide group to form this other species which is equally reactive. Okay, this is still a free radical. So again, this can attack a new monomer and form a longer chain. Okay, so in this way, this is required to initiate the formation of the chain, but this chain can keep on growing in length. Okay, and occasionally what can happen is in this acrylamide chain, we can add this which is called bisacrylamide. So you can see this is one acrylamide molecule. This is a bisacrylamide molecule. It's basically linked by this methylene group in the middle. When we have this persulfate ion, which gives a sulfate radical, it allows bisacrylamide to basically form this. It allows acrylamide to polymerize, and bisacrylamide can form a cross-link between two acrylamide chains. Okay, so you can see if I have one more bisacrylamide molecule here, this is basically going to form a pore, right? And this is the pore from which our particles or molecules in electrophoresis will pass. Okay, so we use persulfate to form this free radical, But we can also use riboflavin and light. So basically, riboflavin in the presence of light will decompose to give a free radical, and that can also be used to form your gel. So in that case, you basically just add riboflavin to your mixture and keep it in front of a light source for a few hours, and your gel will polymerize. Okay, so this is just the basic theory of how to make a polyacrylamide gel. Uh, so the steps in this are you have to set up a gel caster. So basically, we cast this between two glass plates. Okay, the two glass plates in the middle of which a gel will be cast. Okay, so for page, we actually cast two different kinds of gels. One is called a resolving gel, which is where our proteins actually separated on the basis of molecular weight. After we cast the resolving gel, we will add an organic solvent on top. Okay. So what this does is prevent the entry of oxygen into the gel because oxygen will actually mop up all the free radicals in your gel. That will make the polymerization process slow. After this bottom part is polymerized, we actually have to add a different kind of gel on top, which is called a stacking gel. And we'll come to why we need a stacking gel later. And inside the stacking gel, we'll insert this comb. It basically has its teeth, and these teeth will form the wells where you can load your sample. So once this whole thing polymerizes, we'll get an acrylamide gel. When we run this gel, what we have to do is we have to take the gel cassette and keep it in the running tank. There, okay, the anode, if you can see, is actually at the bottom of the gel. Okay, anode is running all the way here, and the cathode is at the top of the gel. Okay, so we are loading the sample near the cathode, and the anode is at the bottom. So what in vertical electrophoresis, what we can do is we can actually use two different buffers. We can fill one buffer near the cathode and one buffer near the anode. But for some purposes, we actually have to use two different buffers. But for most SDS gels, we use a single buffer called a lamellae buffer. Okay, we use a tris glycine buffer to run most SDS gels. After that, once we remove the comb, all these little holes are left where we can load a sample. And then once we apply the potential difference across these two electrodes, our samples will migrate in the electric field. Okay, so when we run an SDS page gel, we mix our sample with SDS. And what that is doing is giving a negative charge to the sample, right? So it will migrate towards the anode. It will migrate downwards into the gel, and it will be separated on the basis of molecular weight. Okay, so here we can see that when we load our sample, we are loading it into this depression, right? So the sample molecules are there one on top of the other. And if we just directly run that into a resolving gel, what will happen is there will be unequal separation of these molecules. So what we first need to do is push all these molecules into a single band that will then migrate only on the basis of molecular weight. And for that, we have to keep this upper part, which is called the stacking gel. So it literally just stacks the molecules together. Okay. And once it is stacked, then we have this mixture of macromolecules, which is then separated because it has to move through the pores of the gel. So the smaller molecules will move faster, while the bigger molecules take more time to travel. Okay, and as a result of electrophoresis, we will get a separation of these based on their molecular size. Okay. So obviously, you can see that the pore size will determine kind of what is the range of molecules that you can separate through this gel. And yes, how you can change the pore size is by changing the acrylamide concentration. So if I have a 5% acrylamide concentration, you can imagine that the pore size will be very big, right? There'll be more gaps between the acrylamide molecules. And so the bigger proteins can move through that. So the range of proteins that we can separate through this is actually quite large, 60 to 350 kilodalton proteins. Okay. And as we increase the acrylamide concentration, the pore size is going to reduce. Okay. So if we have a 15% gel, then we can easily separate molecules which are much smaller. So it's 10 to 100 kilodalton molecules will be separated well in a smaller gel. So then you have to choose the acrylamide concentration based on your application. Okay. So say I am purifying some protein which is 25 kilodaltons. Okay. I will not be able to see anything if I run it in a 5% gel. Okay. Even in a 10% gel, it may not be resolved properly. So it's probably better that I go for 12% gel or 15% gel. Okay, that will be better for my application. But if you have some very large protein complex, which is 200 kilodaltons, then you have to cast, say, a 10% or 5% gel, because otherwise it won't run in the gel at all. This is what an ideal SDS page might look like, where we have these very crisp bands, very sharp bands, and these are the different lanes, and you can see these are all nicely separated from each other. Okay. Once we One primary thing we can do with gels is estimate the molecular size of our protein. Okay, which is why we will often run something called a ladder. This protein ladder is a mixture of standard solutions, which means we already know their molecular weight. Okay, so this is a very standard collection. So myosin here is 200 kilodaltons. So if anything is migrating near myosin, we also know that its molecular weight is near 200 kilodaltons. Similarly, beta-galactosidase is 116 kilo, BSA is 66 kilo, lysozyme is 14.4 kilo, right? So because we know the weights of this standard mixture, we can then estimate what is the weight of any unknown protein band that we get in the solution. For example, we have run some unknown protein and its band has come between 45 and 31 kilodalton. Okay. So just by looking at the gel, you have some rough estimate of what is the size of this protein. But we can actually do a more exact measurement, which is we can use this to plot a graph. So what we know is that log of the molecular weight versus its migration in the gel is actually a straight line. So if we plot log of the molecular weight versus migration, we can then fit a line in this and use it to calculate the molecular weight of our unknown protein. Okay, we'll do a few examples for this. So how can we measure mobility? We just said, I want to measure its migration. How do we measure this? It's literally just measuring this distance. If this is the top of the gel and this is our band, we just need to measure this distance. Okay. This is the die front, which will be the bottom of our samples. So if we want to calculate something called relative front or relative mobility, what we will do is take the migration of this band divided by the migration of the die front. Okay. This is something called relative mobility. Measuring this is very straightforward if your band is just straight. Okay, you get a nice clean band. But oftentimes, if you load a lot of protein in your gel, your bands will look like this. Okay. And then where do you measure? So the best place to measure this is to measure from the top. So you'll measure the top of this to the top of the gel to get your relative migration. Instead, if you've gotten a band which is bulging like this, what you should measure is the middle of the band. Okay? So you can measure the middle of the band to calculate relative mobility. So let's just do a quick example on how we can determine molecular mass. Okay, so let's just go through the question. The following table shows the distance moved in an SDS page by a series of marker proteins of known molecular mass. Okay, so these are all known proteins, transferrin, etc., to cytochrome C with a known molecular mass. This is mass in Daltons. And what they have done is just measured the distance that each of these proteins has traveled. Okay, so this is all known data for us. There is some newly purified protein X which was run on the same gel. Okay, it is showing a single band and it has moved to a distance of 44 millimeters. What is the molecular mass of this protein X? So just if you want to get a rough idea, we can see that 45 millimeters will be between 50 and 38, which means the molecular mass will be between 29 and 36 kilodaltons. Okay, but we can also find a more exact value. So let's do that. So for that, what we'll do is we need to plot a graph. So here I already have the same data. These are all the known proteins with their known molecular weight. and their migration in millimeters. Okay, if you remember a few minutes ago, I said the plot we have to make is of log of the mass. So we will take the log of all the masses. Okay, and what we need to do is plot this in a graph. Okay, so what we have here, this is distance traveled. Okay, and this is log of the molecular mass. Okay, and you can see that this is actually forming quite a straight line. Okay, so which is what we were hoping for. That means that our gel has separated these proteins properly. Now what we can do is find what is the equation of this trend line. Okay, so what I've done is calculated what the line passing through these points, what is the equation of that line. Okay, and we can use this to calculate the molecular mass of any new protein that we are running on the same gel. Okay, so what we know is that protein X, okay, has some molecular mass which we don't know. Okay, but we know that its migration is 45 millimeters. Okay, that means X for this is 45 millimeters. So let's try to calculate what is Y. So I'll just follow this equation. So it is 4.9496 minus 0.0101 into, right? So this is log of the molecular mass of protein X. So how do I get back to what is the molecular mass? I have to take the anti-log of this. Okay. So this is what we've gotten from our equation, 31,267 Daltons. Okay. That is approximately 31 kilodaltons. Okay. What you have to remember is this method has an error rate of about 10%. So when we report this, we will say this is 31,000 plus minus 3,000 Daltons. Okay. So that is, yes, approximately 31,000 is the same answer that we got. Okay. I hope everyone has understood how we did this example. Okay. If you have any doubts, please feel free to ask. And feel free to type in the chat if you have any other questions. Okay. We'll do one more example on this. Okay. This is a very straightforward example. The relative electrophoretic mobilities of a 30 kilodalton protein and 90 kilodalton protein are given. These are standards. So what is the apparent mass of a protein having mobility of 0.62 in the same gel? Okay, so we can use exactly the same method to calculate this. So we have standard 1 and standard 2. This is 92 kilodaltons and this is 30 kilodaltons, right? The mobility of the 30 kilodalton is 0.8. Mobility of 92 kilodalton is 0.41, right? So this is 0.80, this is 0.41. Okay. So first step is to first calculate the log of the molecular weight. This log 10. Okay. So we have to calculate the log of the molecular weight for both of these. Okay, I'm keeping the molecular weight in kilodalton. So whatever answer we get is also going to be in kilodalton. Okay. Next, we will make a chart for this. Okay. Again, we only have 2 points. So this is not the most accurate way of estimating. Ideally, we would have more than 2 points, but that's okay. Again, this is relative migration. Okay, and this is log of the molecular weight. What we have to do is now add a linear trend line and find the equation of this trend line. Okay, so this is the equation that is given by these 2 points. Our, so now let us go back to our unknown point. What is the unknown? It has a mobility of 0.62, but the apparent mass is unknown. Okay, fine. So basically, we know that x is 0.62, so we have to find out what is y. So again, whatever protein x, this is 0.62. So again, we'll use this equation to find what will be the value of y. 2.4754 minus 1.2479, 2.62. Okay, and again we will have to take the antilog of this, which has given us an answer of 50.3. So protein X, according to our calculation, has a molecular mass of about 50 kilodaltons, right? Which makes sense. It should be somewhere in between 30 and 92. So it makes sense that 50 should be the apparent mass of this protein. Okay. So this is how we can estimate size based on an SDS page gel. Again, I hope this is clear to everyone. If not, all the sessions are recorded and all the material will be available to you. There's a link in the coursework itself where you can watch recordings of any of these sessions. Now, sometimes our SDS page gels go wrong. Okay, we would love it if our gels look like this all the time, but they don't. So some of the common errors that can come in our gels is sometimes the bands get smeared. Okay, and sometimes they're not resolved properly. This can happen if we run our gel too fast. Okay, that means we have put too much voltage. The gel is running too fast, and that's why it is smeared. Another thing that can commonly happen is you will see that the gel is run more in the middle, okay, and less at the sides. Okay, it's called a smiling shape. Okay, and this can be caused because there is too much heat which is produced in the electrophoresis process. Okay, so if you run it at a higher voltage, there'll also be more current running through the system. Okay, that generates a lot of power which is released as heat. So if you run a gel, you will notice actually that your tank will get quite hot. And if you run it at too high a voltage, there can be a lot of heat produced, which can cause shapes like this in your band. Okay. Another issue that can happen is if you leave these two side wells empty, what can happen is your samples can actually migrate outwards into these, and it can cause this kind of umbrella shape in your gel. So here the only solution is to load the side wells. Don't leave the side wells empty, otherwise you can get problems like this. Okay, and this, if you have something like this, then you can see that now we can't apply the same relative migration theory, right? Because this has actually moved like this in a diagonal direction. It's not moved straight. So I cannot use this to accurately find any molecular weight. So we have to make sure from our side that the gel runs as straight as possible. Okay. So that is what I had for polyacrylamide gels. Uh, I will also talk a little bit about the other kind of gel we use a lot in the lab called agarose gels. Now, yeah, hi. Okay, so you have to think of a couple of things. So if the voltage is too high, then your gel will run extremely fast. If the voltage is too low, then your samples will start to diffuse. So ideally, we run most agarose gels between 120 to 150 volts. Okay, usually that is fine for most agarose gels. Uh, if within this range you're getting other problems, like you can see the issue in front of your eyes, that your gel is starting to run like this, or your samples are starting to diffuse from the top, then you have to adjust the voltage based on that. So it's important that you know what issues can come so you can diagnose what is the problem. But mostly 120 to 150 volts is good for most polyacrylamide gels. Okay. Okay. So in an exam, ideal. So I'm just using a function in the Excel sheet. Okay. So in an Excel sheet, if you press the equals to button, you will get the option to do a lot of math problems. So I can do sum. Okay, this is just going to do addition. So if I type log, okay, what log is going to do is take the logarithm of whatever input I give here. Okay, so this is just a function within Excel. I'm not doing anything fancy. If you on a calculator also, you'll have the same option for log. Actually, even on your mobile calculator, you'll have the same option. Uh, if this kind of question comes in the exam, all I can say is you will have to try to calculate fast because I mean, it should take maybe 5 minutes maximum, right? Because it's very straightforward. You know how to do it already. Uh, but yeah, I hope this kind of thing doesn't come because it will take time to make the chart and then calculate your value. Okay, so yeah, if you have any other questions, please leave them in the chat. Otherwise, I'll move on to agarose gels. So this is, as I said, this is the second major kind of gel that we use in the lab. So agarose is a linear polysaccharide. It's made up of this unit called agarobioes. Okay, you can see it's a disaccharide, which is just repeating units. So what happens is this forms hydrogen bonds with other agarobioes units, and that forms a gel with pores. And what happens is usually we make 1 to 3% solution of this agarose, and then we boil it. And once we boil it, it dissolves in the buffer, and then as it cools, it actually polymerizes. Okay. The pore size of this gel depends on what is the concentration of agarose that we have used. So again, if we use less agarose, the pore size will be big. If you use more agarose, the pore size will be smaller. We use it mostly for nucleic acid. So you must have heard running a DNA gel, RNA in an agarose gel. It can also be used for large proteins. Okay. So like if you remember here, we mentioned that the protein fractionation range, even for acrylamide, is around 350 kilodaltons. So what if you have some big protein complex, which is bigger than this, right? Then what will you run it? So you can run it in an agarose gel, or you can run it in a mixture of agarose and acrylamide gels. Okay. So 1% gels, as I mentioned, we have less agarose, it will have a large pore size. So we can again use it for some applications such as isoelectric focusing. Okay. Because this pore size is much bigger compared to most proteins. So can move freely through the gel. But DNA and RNA are actually larger molecules as compared to proteins. And so we can separate them easily on an agarose gel. So this is an example. Here we have this horizontal gel electrophoresis. We have an agarose gel. It is, and here we have a positive electrode, negative electrode. And say we load a sample here, it can migrate towards the other electrode. And you can see that the buffer system is continuous. That means both these electrodes will be dipped in the same buffer. So this is a typical agarose gel. Okay, this is a 0.8% gel in which we have run some DNA. Just note that the wells are down here, which means the gel is actually run in this upwards direction. And you can see very nicely the bands are separated, and you can clearly see the different sizes of DNA that were present in this sample. This is a typical RNA gel. Okay. Again, RNA is a smaller molecule than DNA, so we'll have to use a higher percentage of agarose to run this. So this will be 1.5 to 2% agarose gel we'll use for RNA. Another thing you have to consider when running RNA is that RNA will usually have some secondary structure. Okay, it can form like a loop like this. It can form different kind of knots, etc. So usually, if you want to run an RNA gel, you will have to denature the RNA, which is why if you've ever heard of using like a formamide gel or anything like that, that is for denaturing the RNA so that you can separate it nicely on the basis of its size. Okay, so the other major application of DNA gels is in sequencing. So you might be familiar with this in old-time sequencing methods, like the basic sequencing methods. The way that the sequence was read is by running basically different DNA fragments on the gel and then just reading off the bases one by one. Okay, so we'll just do an example of how to read a sequencing gel. Okay, so these are each of the 4 nucleotides. We will assume that a radioactive nucleotide has been added to the end of each fragment. Okay, and so this fragment is basically a fragment ending in radioactive G. This is the fragment ending in radioactive T. So on. And remember that the smaller molecules will migrate first, right? So actually our DNA sequence is starting from here. And this is the same DNA sequence with 1 nucleotide added. Okay? If we added 1 more nucleotide, so it's become bigger, so it's migrated less. So if you want to read the DNA sequence, you have to start from down and then move up. Okay? So let's just try reading this DNA sequence and see what happens. So we'll start with this normal gel, this first gel. Okay. So here we can see that the bottommost band is corresponding to G. Okay. After that, the second band is T. The third band is G. Right. And again, after G is C. T, G. T, C, T. C, C, T. G, C, C. Right. Then again, this will come back to G, A, C, and A, A, G. Okay, this is how we can read a sequencing gel from bottom to top. Okay. So what, if we just go back to the question, what it is asking is, there's sequencing gels for different variants of hemoglobin. It's asking what is the amino acid change in each variant. So similarly, we can read the sequencing gels for each of the variants. Okay, I'll just do one more. This Chong Queen, whatever this is. Okay, GTG. This is CGG. Okay, here immediately you can see a difference between these two gels. Right, so this was CTG, this becomes CGG. So immediately this is where some mutation has taken place in this variant. Right? Again, if we continue reading, this will be TCT, CCT, GCC, GAC, and AAG. Okay, similarly for Karachi, TG, CTG, TG, CCT, right? CT, CCC, GAC, CC, right? CC. Okay, so here we have read what is the sequence of the hemoglobin gene, of some region of the hemoglobin gene for each of these four variants. So now I think it's very obvious that here there's one mutation here. The Karachi mutant has one mutant here. Swan River has one mutant here, right? And if you want, we can then try and see what this mutation is. So in Chongqing, so CTG has become CGG. So CTG, which is a leucine, has become CGG, which was arginine. Okay, so leucine has become arginine. Okay, again same thing here. GCC has become CCC. So what is GCC? GCC, which is alanine. This is alanine has become what is CCC? Proline. Okay, this is a big change. I mean, alanine is a tiny amino acid. Proline is a large circle, like circular amino acid. So this is going to make a big difference on the protein structure. Okay. Next, what is this? GAC. GAC aspartate. As has become GGC. Glycine. Okay, and again, this means the charged amino acid has become an uncharged amino acid. So again, this will have a big change on the nature of the protein. This is all information that we got from the sequencing gel. Okay, so electrophoresis is useful in everyday life beyond what we do in the lab. So quickly, I'll just go through a couple of other concepts that maybe sir didn't focus so much on this week, but they're also interesting to know. So one of them is called isoelectric focusing, and this is basically separating molecules on the basis of their pH. Okay, everywhere else we are separating them on the basis of charge. This is going to separate them on the basis of pH. So what happens is we need a gel in which you have to establish a pH gradient. Okay? This is done by using something called an ampholyte. So when we add a mixture of ampholytes in an electric field gradient, what will happen is that they will migrate to different pHs, to different places in that electric field and establish a pH gradient. And then when we add our sample in this, what will happen is the protein will migrate in the electric field until it reaches its isoelectric pH. At the isoelectric point, the positive and negative charges on any molecule are equal. So net charge is zero. So it will no longer move in the electric field. Okay. So basically, based on this isoelectric point, we can now separate the molecules. So here, proteins migrate to the isoelectric pH, the location at which they have no net charge. Okay, so this is a different way of separating molecules. What? Yes, this is just a different way to look at it. We have an electric gradient, which is developed by an ampholyte solution. Then we are adding our protein sample and applying the electric field, and the proteins distribute according to their PI value in this gel. Okay. Well, how this is useful is in something called 2D electrophoresis, where we have this isoelectric focusing, so that we call the first dimension. What we can do is take this and basically add it to an SDS-based gel. And now our samples are already separated on the basis of PI. So we can now run the SDS-based gel as normal and they will now separate on the basis of mass. Now we've separated the samples based on two different ways, based on their pi as well as their mass. So now if we had samples which had the same mass, two different samples with the same mass, but different isoelectric points, we can see them as two different spots in this gel. So here, this is what a typical 2D electrophoresis gel will look like. So here you can see spots corresponding to more than 1,000 different proteins. Okay, this is some E. coli cell lysate, but this is not a level of separation you will ever get through a normal STS page gel. Okay, so here basically everything you're seeing in the same line, these are all proteins with equal mass but different isoelectric point. And everything in the same vertical line are proteins with the same isoelectric point and different mass. Okay, so this is something called 2D electrophoresis. Another form of electrophoresis is called capillary electrophoresis, where we move the samples through a capillary. Okay, where basically we apply the sample here and then we apply an electric charge across this capillary. The sample is forced through the capillary where we have a detector. Okay, so basically samples pass through the detector one by one as they move under the influence of this electric current. And here the main parameter that we monitor is called the migration time, which is how much time it takes to come to the detector. And that depends on the length of the capillary, obviously. If the capillary is long, it will take more time to come. The electrophoretic mobility. So if it is electrophoretic mobility is more, it will move faster. And the applied voltage. So normally in a capillary electrophoresis, we'll apply a very high voltage, like 50 kilovolts, and use it to separate some very small samples. So this is just an example of what a capillary electrophoresis trace can look like. Okay, what they've separated is 5 different peptides. Okay, peptides. Can like imagine how small that is. 5 separate peptides who are just different in 1 or 2 amino acids. Okay, if you just ran this on an SDS page gel, you would not see any difference between these. But what you can see is all of these have slightly different charge. Okay, all these are slightly different charge and slightly different size. And when we run it through capillary electrophoresis and use a UV detector, so basically this detector is detecting UV absorbance from these peptides. Then they have different basically migration times. So this is migrating first, and this is migrating last. Okay, and we can basically resolve all of these quite well. Okay. Any questions on this part? Sorry? Yeah. No diffusion. Sorry, no diffusion will happen. Yes, there will be some degree of diffusion. I mean, this is a problem with all electrophoresis, in that we are getting some level of diffusion, and so that's why you don't get a crisp band. Okay, but in 2D it's not a bigger problem in 2D compared to uh, other basically gel techniques. Okay, okay. So basically, we have done isoelectric focusing here. So all of these different bands have different pi. Okay, so that means this band has something of, say, let us say pi of 5. Okay, is in this dark band. So all of these have the same pi. But now when I'm running it through an SDS page gel, they will now separate on the basis of weight. So this band is now separated into these 4 spots. But since all of them were originally in the same band, that means all of them have the same pi. Okay, they just have different molecular weight. And as we know in an SDS page gel, they're separating on the basis of molecular weight. So things which are running at the same level have the same molecular weight. So these two are running at almost the same level. right? Which means they must have the same molecular weight. But because they're at two different spots, they must have different pi. Okay, you have to remember in 2D, it's not like we're loading different samples here, here, or here. We're just taking this isoelectric focusing gel and just directly placing it on top of this SDS gel. Okay, there is no stacking gel or anything. We're just directly running it. Okay. Again, if you have any questions related with this part, feel free to ask. Otherwise, what I was going to do is I need to load an agarose gel. So what I was going to do is maybe take you through that. And if you have any questions related to the practical session, again, feel free to ask me. Yeah, I'm just joining from my phone so that I can take you to the gel loading area and we can load an agarose gel. Okay. Okay. I'll just stop sharing here and yeah, I'll turn off this video. I think you can see me here. And yeah, I'll just take you to collect my samples. So I need to load some PCR samples. I'm just getting those out of here. Okay. Um, just give me a second to put on some gloves. Okay, safety first. So most agarose gels contain lithium bromide, which you should not be touching with your bare hands. Gloves. We'll now go to the gel area where it's an agarose gel already cast. Okay, and what we'll do is prepare the samples for loading. So for loading, we need a loading die and a reference standard. So probably easier like this. This is what a reference standard will look like. Okay, so this says gene ruler 1 kb. So this will tell you about what kind of bands you expect to see in the standard. And the other thing we need is a loading die. This is 6x DNA loading die. This is basically bromophenol blue, glycerol, and water. Okay, it's not anything fancy. But this is a 6x die, which means that we have to dilute it when we add to our sample. So what will happen is our sample is 20 microliters. So we will add 4 microliters of loading dye to our sample. So final volume will be 24 microliters, of which 4 microliters will be the loading dye. So that's 1/6 of the sample volume. So it is diluted to 1x. I'll just set this up here. So I hope you can see. I hope you can see. So again, I'll set this to 4 microliters. Okay, and basically I just have to add dye to all our samples. Okay, add it to this now. Okay, now I've added loading dye to all my samples, which means they're ready to load. I will now take my gel and put it in the running tank. So, like we discussed, this is the running tank. Okay, so yeah, this is the negative electrode. Okay, this is the positive electrode. So we will run the gel like this. Okay? Because DNA has a negative charge, it will run towards the positive electrode. Okay? This is connected to a power pack, so I need to turn this on. Okay. Here you will see options for to set voltage, current, time, etc. So we'll come back to this. Okay. First, I need to release my gel. It's already set, so I have to take it out. So, like we discussed, this is a vertical slab, right? It's a horizontal slab where this comb is used to make wells. So this I'll just put directly in the tank. Tank is already full of running water. And then what we have to do is remove the comb. Okay. Now I hope you can see that these are a series of wells. This is where we're going to load our sample. Okay. So I'll just set the pipette to appropriate amount. I'll get a tip box and I'll start loading. So when we take a sample, you can see the dye is kind of at the bottom. So we'll first mix it properly. Okay. And now we'll load this in the gel. So I'm just pushing the tip into the well and releasing it. So I'll continue this for all the samples. When you load a gel, it's very important that you keep track of what is the sample order. Otherwise, you won't be able to interpret what is the result of your gel. I've casted well with 8 wells. Okay, so that means I will load 7 samples and one ladder. And last sample. Okay. Now I just need to load the ladder. I know the ladder is fairly concentrated, so I will load 3 microliters of ladder. Okay, I'm done loading. So I'll show you what the. So you can now see that the wells are full with the dye, right? So you can easily see that the samples have been loaded into the well. So I have 7 samples here, and the last well is my ladder. So now we have to set the voltage and so on. So you can see already someone has set voltage for 120 volts. I can run it here. This is not a problem, but it will take some time to run. So I'll just increase this. Okay. I'm happy with these parameters. So I'll start. So again, this is trial and error. So you will have, depending on the size of the gel, your power pack, and so on, you'll have to try a few parameters and see what is best. But generally, like 100 to 140 volts is the range we run inside. If you increase the voltage, it will run faster. But then again, you might also get like other artifacts, like it might run in a slanting direction and all that. So this is just trial and error. Okay, so here it started, and you know it started because you can see bubbles coming out of this electrode. Okay, right? You can see bubbles at both sides, which means that this is running. Okay, and you will see that the sample is starting to come out of the well, right? Because it's starting to move towards the positive side, right? So we'll let this run for maybe 10-15 minutes. In that time, I will keep the ladder back. Okay, ladder is very important. You should keep track of it and take care of it. So I'm just keeping this ladder back. Its role is it's giving us a reference point. So otherwise, we can run the gel and we can see the nature of the DNA bands, but we won't be able to comment on the size. But the ladder has known molecules, has known size. So we can compare with the ladder and get an estimate of the size. So what's the ladder we will use? Usually we will use only one. Uh, Most labs will have one DNA ladder and one protein ladder. But yeah, depending on, so if your sample is very big, so your sample size is say 600 base pairs. So your ladder should have that range in it. If your sample is 100 base pairs, then even that should be there in your ladder. So you have some ladders like 1 kb ladder. So bands are at a distance of 1 kb. So if you have a very small sample, say I have my 100 base pair sample, I won't run it with the 1 kb ladder because it won't get any reference from that. So you have to run it according to the nature of your sample. So I've just loaded it along with the sample. So it will run at the same time as the sample. When we visualize the gel, we can see exactly how it looks. Okay? Yes, that is so that is always a temptation that we will run with at a very high voltage. But another thing that will happen if you as you increase the current, it will run faster, but it will also start generating more heat. Okay, and you don't want it to start denaturing your sample or anything, and you don't want it to start affecting the electrophoretic process itself. So yes, I can run it at 200 volts. Maybe it will run extremely fast, but that won't be good for my sample. Usually, yes. Yeah. So for most samples, okay. So there are some specialized kinds of electrophoresis, which I think Sir will discuss in the next week. Things like pulse field electrophoresis and so on, which we use for extremely big DNA samples. Okay, I'm talking like like mega base pairs, etc., and so on. So for that, we use different kinds of methods. But for most, if you're running like a plasmid, you're running some small DNA, 10 kb or so on, we run up to 140 volts. Yeah. And the kind of gel that you cast, obviously, you can cast different kinds of gel depending on what your sample is. So maybe this is better. So here, this is the well I used today. You can see there are 8 pots to put samples. Okay, that means I can load 8 samples, and the volume of each well is determined by this. What is the size of this? Basically, this tooth. So I can load maybe around 50 microliters of sample in this well. But if I have more sample, so the volume of my sample is more, then what should I do? Then I can basically find a different comb. Okay, so if you can see this comb, this middle well is extremely big, right? If I have maybe 1 ml of sample, then I will use this instead to cast the gel. Okay, so I can load a much bigger sample here. If I only want to run this gel for a small amount of time, say my DNA size is very small, so I only want to run the gel for 5 minutes or something. I know it will get separated in that time. Then I can make a short gel. Okay, this will run fast and it will take less agarose. So if I know my sample is small, I don't want to separate it that much, I can use something like this. If I have a lot of samples, right? We just saw that here I could fit 8 samples. But what if I have 20 samples? What should I do? So you can make cast a bigger gel. Okay. So just to compare, this was the size of original gel. This caster is almost two times as big. So I can run that many more samples. Okay, and correspondingly you can get a much bigger comb and cast many more wells. Right? These are all things you can choose depending on the nature of your sample. Okay, so we'll let this run for some time. You can see maybe it's starting to separate. I've found different casters for casting agarose gel. Okay, for sure we can discuss polyacrylamide gels as well. So polyacrylamide gels are cast in a vertical manner. Okay, so this is the casting apparatus we have. Your lab might have a different apparatus, but it is cast between 2 plates. Okay, so one plate will look like this. Uh, I don't know. I probably can't see. Okay, so you can see that this plate basically has this region which is raised from the plate. So what we do is put this other plate on top of it, and you can see it's now formed a gap. Okay? So this gap is where we will cast our gel. Okay, and the size of the gap is mentioned here. This is 1.5 mm gel. Okay, so this gap is 1.5 millimeters. So basically, what we'll do is we'll put our plates like this. We will secure it, and in this gap, we will start pouring our gel mixture. And I'll just show you what a comb looks like. So this is our agarose comb. Remember, this is what a comb for SGS page will look like. Okay, these are all teeth that will form the wells, and uh, this will fit nicely in this gap here. Okay, it's just going to fit like this. Okay, so what you do have to do is look carefully at this. You can see here is written 1.5 mm. 1.5 mm, 11. So 11 is telling you how many wells are going to be cast. And 1.5 mm is telling you for which gel we should use this. So this is to be used for 1.5 mm gel, so we'll use it with this plate. The other kind of comb that we have is called 1.5 mm 15. So this is going to cast 15 wells. But you can see that the volume of each well will be less, right? Because this is much thinner. But if you have many samples, you can use this instead. Also, like I mentioned, this was 1.5 mm gel, which is what we use usually because it's quite easy to handle. But if you want to run your gel really fast and you're very confident, the other thing you can use is this, which is a 1 mm plate. Okay? So that just means that when we cast the gel, this distance is less. Okay, so this is 1 mm separation between these two. This will cast a very thin gel. Okay, and it will run very fast. Okay, for the 1 mm gel, you have to use a special 1 mm comb. So this is a 1 mm comb. This is 1 mm 15. Okay, so this you use with a 1 mm gel. If you try to put this in a 1.5 mm thing, you will see that it is loose. Okay. And yeah, what I can show you is some SJ space gels that I ran yesterday. Okay, we can just talk through what is in these gels. So this is gel that has been run. It's been stained with Comasi dye. So all the protein bands will be blue. And then it's been de-stained. Okay. So here you can see, here on the right side, the rightmost lane, this is some large mixture of proteins. This middle lane is a standard or reference, which is called a ladder. So these are all bands of known molecular weight. So since I already know what is in this ladder, I can tell you this is 25 kilodalton, this is 20, this is 15, this is 10. Okay. Above it, we have 37, 50, and 75. Okay, these are all large molecular weight proteins. Okay. And I was running this gel to look at this protein that I purified. So these are all fractions that I got from eluting my protein. So here you can see my protein is around 25 kilodaltons. Right, this is my known standard. So this is 25 kilodaltons. Okay, and I can see that in the first fraction, I have a some protein. the amount is increasing. So in the third fraction, I have the most protein. And then 4, 5, 6, 7, I have this. And then the rest of the fractions, there's no more protein. So I know if I want to get maximum protein, I should take maybe 2, 3, 4. I should take these three fractions and pull it. Okay. So this is what an SDS page gel might look like after you're done. Like I mentioned, this is Kumasi staining. So that is the standard way to visualize gels. It has a sensitivity of maybe 0.3 to 1 microgram. So you need to have that much protein in your gel. If you don't have that much protein, you can use a different kind of staining method. So the other one we use in our lab is called silver staining. Okay, so that can detect protein at nanogram level. So this is what a silver stain gel will look like. Okay, again, this is the ladder. It's the same ladder. Okay, but now if you look at the molecular weight bands, this is 75, this is 50, this is 37. So whatever molecular comes between 50 and 37 kilodalton. Okay, and it is, you can see it's a very clean fraction. There's nothing above or below this. Okay, so very clean protein purification of some protein. Okay, let's go back and see what our agarose gel is doing. Okay. I hope you guys can see that there are two blue bands in each gel, in each well. That is because the loading dye is actually a mixture of two components. There's bromophenol blue and xylene cyan. Okay, so that is giving us these two bands. So this lower band will be around 200 base pairs. This is around 1 kb. So this is giving us an idea of how much the DNA has traveled in this gel. Okay. The product I want to see is around 2 kb. So I need to let this run for some more time. Okay. So we'll come back to this later. Okay. So in the meantime, what we can do is I'll talk you through an image analysis software called Image Lab. Okay. Which I think Sir had shown in his presentation as well. But we'll just talk through the Image Lab, and we can talk through what maybe good gels look like and what a bad gel looks like. Okay. Yeah. Okay. So yeah, I'll just present my screen so that we can look at something which I found quite amusing. It's called the STS Page Hall of Shame, which is showing some gels which didn't work out the way that they were expecting. So you can also see this and think about maybe if you're seeing something like this in your gel, it can help you diagnose what went wrong. Okay. So here you can see some really strange gels, right? Something has gone really badly wrong in this gel. So here they're saying it's a smeared gel. They're saying it is probably due to unevenly poured acrylamide, right? So that means when we were making the gel itself, we made some error, right? This again is smeared because there is too much protein. So all these bands have literally just smeared together in this gel. Okay? Again, here there's just too much protein, so it's just become a big blob. What else? Um, Another thing that you may commonly see in the gel is this. You'll see streaks running through the gel. Okay? And this can become because there might be some minute particles or imperfections within the gel, and that has caused the protein to become a streak like this. Okay? So just an example of what streaking can look like in a gel. So this is very common. Okay, so you will probably encounter this in your gel at some point of time. It is probably due to some fine particulate mixture that was there in your sample itself. In this gel, you might also notice like this top is very uneven. Okay, usually we want this to have a straight top like this, but this is very uneven. And that is happening because it wasn't set properly. So that means that maybe they didn't add isopropanol or any organic solvent on top of this while casting the gel. And that's why they've gotten this very uneven surface. Okay. And this can also cause because of this uneven surface, the protein is running at different levels. Okay. And that's why we can get streaking like this. Another problem we can have is you loaded your gel, but you can't see any band. Okay. So that is when you see a gel that looks like this. Okay. There's something here, but all the other bands are just too light for us to make out. Okay. So this can just be maybe you didn't load enough sample, or you loaded the sample but it came out of the well. Okay, like these are all issues that can occur, and you should watch it. What else? Now, ideally, we want to run the gel so that all our bands separate. But if we don't run it for enough time, then we won't get enough time to separate properly. And so you will see that it's just stopped halfway through the gel. So we're not going to get good separation of these proteins. Okay, so it will be hard for us to analyze anything here. So here all the bands are just clumped together here. You can imagine it'll be much easier for us to understand something if they just run the gel all the way to the bottom, right? So again, when you're running the gel, you have to make sure that you run it all the way to the bottom, so you're getting the maximum amount of information from your gel. Okay. And sometimes the gel can just look strange, right? Something very strange has happened here. This, everyone is asking what happens if you run it high current, if I just increase the voltage, you might get something like this. Okay, the gel is just overheated. The voltage is too high and the gel is just overheated. Here, this gel is distorted in some strange way. They think that it's because they loaded the sample, then waited for a long time, and then they ran the gel. And so there's some diffusion that has gone on, and that's why it is distorted like this. Okay. Here, what they're getting is actually contamination from the buffer itself. So here you are seeing this purple background in the gel. That is because there was some protein in the buffer itself which is stuck in the gel. And no amount of destaining is getting rid of it. Okay. So these are just some examples of things that can go wrong in your gel. And if you do, you can always come to this page and see if you can see anything similar and have a look at what went wrong. Okay. And yeah. Now, what else we can do is maybe look at an analysis software called ImageLab. Okay, so just give me a minute. I will get out of this. Okay. So here I have some gel images. So earlier in the session, we had calculated things like molecular weight based on first we had to draw a plot, and then we had to calculate what is the log value, and then draw a line, and then calculate from the equation of the line what can be the molecular weight of our protein. But all of these things can be done by software, and they are done by software. Okay, so I'll just talk about an image analysis software called ImageLab. Okay, so I'll just show you that this is an image of an SDS page gel. Okay, this is the standards, as you well know. And these are some bands. And I want to know what is the weight of this protein. Okay, so I can say roughly that maybe it would be between 10 and 15 kilodalton. Sorry, 15 and 20 kilodalton. But I want to know the exact weight. So for that, we can use a software called ImageLab. Okay, this is a free software by Biorad. So if you want, you can go and download it. Uh, but what we can do is open this in ImageLab and then analyze it. So first thing we'll do is just find the area that we want to analyze. So this is all too dark. I won't be able to get any information out of this. So I'll just restrict myself to this much of the gel. Okay. So this much of the gel I want to analyze. Then what we need to do for the software is define which are the bands that we want to analyze. So first we'll ask it to automatically detect which are the lanes in our gel, where it's done quite a good job. But we can just slightly adjust this so that it properly captures the lane. Okay, there is nothing here. This again, this band is not in the middle of this lane, so I'll just move this a little. Now it's in the middle. Okay, so now I think all the bands are there properly in some lane. Okay, it's detected 6 lanes, and the third lane has a ladder. Okay, that means we're ready to ask it to detect our bands. So automatically it has found which are the bands in our ladder and which are the bands in our sample. Okay. So now did we want to do? So like I mentioned, we want to analyze what can be the molecular weight of this protein. So we'll use this called a molecular weight analysis tool. So here, okay. So here you will see a box called standards. Okay, and that refers to which standard we have run over here. So we just see what standards they have. So these are all protein standards because this is a protein gel. Now I know which standard I used, which is Biorite Precision Plus. Okay, so depending on which reference you have used, you have to input that. You can see all of these will have different range of molecular weights, low range, high range, broad range. So again, depending on which standard you've used, you have to set that as a reference. Okay, standard lane is 3. And now it has detected, you can see here it has added the molecular weight for each part, each band within this standard. Okay, so now it has become much easier for us to see that this band is between 20 and 15 kilodaltons, right? Okay, now, but what if I want to find the exact molecular weight of this? So that I can do by going to this thing called analysis table. I want to know what this band is, so I'll go to the 5th lane, and that in which it is detected this is the 6th band. Okay, it is detected the middle of the 6th band. So if we go band number 6, okay, and we just go, it's detected molecular weight of 16.1 kilodaltons based on this. It would seem sensible. 15 is here, so it should be near 15. So it's about 16 kilodaltons. Here you will see this value called relative front, which is the same migration that we were discussing in our previous example. So this is just the amount that this band has traveled divided by the full length of this gel. Okay. So this is how we can detect the molecular weight of any band using our reference band, okay, in this software. So this is giving us actually a more accurate representation than the method we were using earlier. Okay. So again, if I want to say what is this, I don't know what this molecular weight is, but I want to find out. Again, I can go to analysis table. This is lane number 6, and this is band number 9. So I go here, giving a molecular weight of 43.1. Okay. So in this way, I can analyze the bands that I've gotten in this gel. Okay. What we can do now is go back to our agarose gel and see how it's doing. Okay. So should have run by now. So I'll just take it out of the tank and we'll visualize it. let's just see how our agarose gel is doing. Okay. Uh, you can see it is now run for almost 30 minutes. So yeah, it's run a fair amount. Okay, so this is where we started. This is the diaphragm. Okay, so it's run that much. So which I am happy with. Okay, that's enough for me to see my band. So I'm going to stop this. Okay, I'll just wait for the voltage to go to 0, and then we'll remove this and see what is there in our gel. Okay, so hopefully our PCR worked and there is something in our gel for us to see. So I'm just going to pick this up. Okay, just get rid of excess buffer. I'm going to take this over to a gel emitter. Okay. This is the gel emitter. You might have seen the Biorad imager in Sir's actual lecture, but this is the gel emitter that we have. So don't drop it in. Can be fragile, but okay, we're just going to put it in there and close this. Okay. And now I will use just ask the software to load, which will take a while. Okay. So this is just using white light to illuminate it, and you can see the outline of our gel. Okay. So the wells are here, and hopefully our DNA will be somewhere here. So to see the DNA, we have to change from white light to UV light. Okay. We'll turn on the UV light. I'll just increase the exposure a little so it's easier for us to see. And I'll capture. Okay. So this is what a gel is looking like. Okay. You can see that it's these are all bands. Okay. In our this is a PCR product basically, and this is a reference standard. Okay. So people had questions about the reference standard. These are all bands of known molecular weight. So this is 1 kb. Okay. This is 3 kb, 6 kb, 8 kb, 10 kb. So this gives me an idea of what size of product I have gotten. Okay. So for example, this is between 1.5 and 2 kb. So, and this is a very bright band. Sorry, this is a very bright band, right? So this is an example. Obviously, I've gotten the PCR product. Okay. Here I've gotten a slightly bigger product, and here I haven't gotten any product. Okay. So I'll just capture this image, right? Now, obviously, this in this image the bands are light and the background is dark. What is can be easier for us to analyze is to invert it. Okay, so now it's easier for us to see the bands against the background. Okay, so we can just play around with intensity, right? But I think the original image was fine. Okay, and then we can save it. Okay, so that's what agarose gel electrophoresis basically works. Uh, you might be wondering what is all this dark stuff still stuck in the well. Okay, this is actually probably genomic DNA. So this is probably from the template that I used during PCR. This is all genomic DNA which is too big to migrate. So it is stuck in the well itself. Okay, remember the 10 kb is here, and this is much, much bigger than 10 kb. Okay, so that's why it's still stuck up here in the well. Okay, so I will just rescue my gel, get it out of here. Uh, Always remember to clean the imager. Okay, be nice to people. So you give them a nice clean imager. You get some tissue. We just wipe off this region. And now we want to discard our gel. Okay, our gel contains ethidium bromide, right, which we've used to visualize the DNA, which can be carcinogenic, certainly toxic. So gel never goes in the regular trash. It never goes in the sink. It goes in a specialized discard for gels itself, which should have a biohazard bin. Okay. Yeah. We just rinse the caster. Okay. Turn off the gel apparatus. Close the tank. Okay, because you can reuse the buffer. And we're done with agarose gel electrophoresis. Right. I hope this was clear to everyone. A fairly straightforward gel that we ran. Just wanted to see if I had a product of 128 kb, and I got it in one of my samples. Okay. So I'll, I'll just show you how to do one more thing, which is how to quantify bands using ImageLab. Okay, and then we'll wrap up the session. Okay, yeah. We'll just take a look at one last gel. Okay, I'll look for a nice gel. Huh. So this is a gel where someone has done like a lot of samples. So she's actually used one comb over here and another comb over here, right? So she's done two sets of samples in the same gel with a set of ladders. Okay. And with this ladder, obviously we can find out what is the size of these bands. But one more thing we can get is an idea of how much DNA is there in each band. Okay, so I'll just take a minute to explain this. So when you get your ladder, it will come with a document like this, okay, where it will talk about what is there in your ladder, what are the bands. Okay, so here it tells me what are the bands. Like I said, the 1 kb band, 3 kb band, 6 kb band, etc. etc. But here you will see another column saying nanogram per zero point 0.5 microgram, which is basically telling you how much DNA is there in each of these bands. So I know that this is 70 nanogram, this is 70 nanogram, this is 60 nanogram. So I can use this to find the relative intensity of my band, and so I can get an idea of how much DNA is there in my band. Okay, so we'll just try to do that using ImageLab. So same deal, I'll just open the image here. I will crop it for the region I want to analyze. So let's look at this. It's very straightforward. We have one ladder, four wells. In these two wells, we have product. In these two wells, we don't have any product. Yeah. Again, we have to ask it to first detect the lanes, which it has done, no problem. Both of these bands are in this lane. Ladder, it's detected properly. It's not detected this lane, but that's okay because we're not analyzing anything there. And we'll ask it to detect bands. Okay, here you can see it's missed a lot of bands. Okay, so it detected these two. So what I can do in this case is just add all the bands that it missed, right? Because software isn't perfect. Uh, Sometimes the software is very far from perfect. But we can just add all the bands that we can see in this gel. Okay. Okay. So now I have added all the bands that I could see in this ladder. Okay, again we'll select, now this is a DNA gel. So you have to select a DNA standard. Standard lane is 4. What is it done? Okay. So here we can see some of the limitations of the software, where it basically has now decided only the bands here count, and it has not used any of these bands. So, but okay, that's okay. We'll try to do a quantity measurement instead. So I'll select this and I will say what is the known quantity of DNA in this band. So for example, if it's 70 nanograms, I'll add 70 nanograms. Okay, nanograms. Here is saying the other bands are 30 nanograms. So okay, we'll try that. 30. Okay, see again, it's automatically detected that this upper band is around 70 nanograms. Okay. Okay, so now the computer's got an idea of what the band looks like. So now what we'll ask it to do is, now what is the amount of DNA in this band? Okay, so what it's done here, here we see absolute quantity in nanogram. Okay, based on the reference we've given, how much quantity as a calculated is there in our band. Now it is saying that 3000 nanograms or 3 micrograms of DNA in this band. Okay. Similarly, if we look at lane 1, it is saying that 2489 nanograms. It's 2.5 micrograms of DNA in this band. Okay. So it has detected the amount of DNA that is there in each of these bands based on the reference that we have given. And this can be very useful for cloning or PCR product to see how much product you have gotten from your reaction. Okay. So this is just an example of some things you can do in the image analysis. Uh, so we discussed how to use the molecular weight analysis tool, how to use the quantity tool. Annotation tool is just allows you to add a text box and say what is in the lane. So you can just annotate what is there where. And this volume tool is just another way to measure the quantity within each band. Okay. So yeah, in this way you can analyze any number of gels. So uh, we can take a look at some more gels. Yeah. So in this gel, you'll see that there is a lot of smearing, right? Here there's basically no product and there is some smearing of the product. Okay. So maybe this gel was run too fast, or maybe this gel wasn't made properly. Okay. What else do we have? This is a very nice gel where you can see PCR product relative to the ladder. The PCR product is actually so much product the detector is saturated. Okay, that's why it's red in the middle. It has gone above the detector limit. This is the gel we just analyzed. Uh, this is what a bad PCR product looks like. This is just a smear. You can see there's no clear band as such, it's just a smear. Okay? That's because there is no proper amplification of any one product. And here again, you'll see that when we have cast the gel, we've cast a small well for the ladder, because we'll only use a small amount. But we've cast a big well for this PCR product because we have bigger volume. So these are all ways in which you can make your gel work for you. This is an example of an SDS gel in which we have not loaded enough sample. So the bands itself are not visible here. Okay, the marker is like the standard is run very nicely. You can see the bands very cleanly, but you can't see any sample because we just don't have enough protein. Okay? Yeah, good. I may had a question. Oh, oh, I'm so sorry. So, okay, so I'll just quickly go through that again. Okay, we'll just use this image. Uh, we will crop it to the region we want. Okay, crop. We will auto detect lanes. Done a good job. We'll detect bands. Okay. You'll see it hasn't detected all these faint bands. So those we'll have to add manually. Okay. Now, molecular weight analysis we know how to do. We'll just show how to use quantity. Okay, for that we have to add a reference band. So I will select what is a known band. Say this. I know this is 60 nanograms. So unit nanogram. What other? I know this is 70. Okay. This is 30 and it's automatically detected it as 25. Okay. So now we have added 3 reference points. We'll see if it can detect how much DNA is there in this. So we'll go to analysis table. This is the second band. And here in absolute quantity, it is showing how many nanograms have been detected. So this is showing 550 nanograms for this band. Okay. So this is just one way in which you can use this to find the quantity of DNA in each band. Okay. Yeah. I'm very sorry about that. I'll just go back to some of the gels I was showing. So this was my SDS example where there's not enough protein, so you can't see any bands. You can see the ladder, but you can't see any of the bands. This here you can see some smearing. So there must have been some maybe particulate matter in the sample itself, which has caused the smearing of the sample, right? Here you can see this is a PCR product which hasn't amplified properly, which is why instead of getting a sharp band, we're getting this full smear in both the wells. Okay. This is the gel we just analyzed. You can see it's a very clean gel where we're getting one sharp band, and this faint band you're getting at the bottom is just primers which are not used in the PCR reaction. Okay. But yeah, that is what I had planned for today. Again, if you have any questions, floor is open. Feel free to ask, and I'm happy to answer. Yes, hello? Sure. Okay. So when you use a ladder, it will come with a, a document like this. Okay, a user guide. Okay, we'll tell you the details of the ladder, and it will have this region. Okay, we're showing which different bands are there in the ladder, what is the molecular weight in each band, and what is the amount of DNA which is there in each band. Okay, this information will be given with every DNA ladder. So you know how much ladder you have put in the gel. So according to that, you can calculate how much should be there in each band, how much DNA should be present in each band. Okay? So, I mean, usually what happens is what we do is we actually cut out the band in the gel and we purify the DNA from the band itself. So then we can measure the concentration of DNA that we have gotten. This is just like a preliminary check to see how much our PCR reaction has worked or something like that. Of course, it can. So uh, that's why you have to be careful not to let it heat too much, especially if your sample is sensitive to temperature and you allow your run it at a very high voltage, a lot of heat will be generated. Okay, so even in the gel that I just ran, when I took out the gel, it was hot. Okay, and that's because I ran it at 140 volts. If I had run it at 100 volts or something, it would have been a more gentle run and less heat would have been generated. So, of course, this is something you have to be careful of when you run your gels. There is no temperature regulator as such. So, it will just run at constant voltage. What if you are very concerned about this, you can keep your gel running tank in ice. So, not here, but when we do something called Western transfer, then usually we will run it in ice because a lot of heat is generated in that process and we don't want it to affect our proteins. So, yes, in the tank itself, there's no temperature regulator as such. You can leave the tank open, so then there's some like degree of heat loss, but otherwise there's nothing to regulate the temperature. You can, you can, absolutely. But it's just, if you're running a gel every day, it's just a pain to get ice, keep it, and so on. That's the only thing. No, so actually, so the software is from Biorad. So they have actually only kept the Biorad ladders there. So if you're using a ladder from some other company, you will have to add it manually. So you'll have to manually select the lanes and say that this is corresponding to this KB. The preloaded ladders are only from their own company. Yeah. So no, no. So if you're talking about the bulging in the band, no? If when the band is becoming, yeah, it's due to sample amount. If you have too much of a certain sample, it will bulge. The band will bulge. Nothing you can do. Yeah. So you're talking about the buffer. Yeah, the buffer which is used for running the SDS page. Yeah, yeah. So the buffer we use is a tris glycine buffer. Okay. So no, no. So it is a very standard buffer that we if you say SDS page, your buffer will be tris glycine SDS. Okay. But there are other things you can do with this, like native page, urea page, acid urea page, tricine page. Okay, these are all different variations that I think Sir will discuss in the coming week. So these all have different applications. So, for example, if you don't want to denature your protein, you will use a native page, and that there is no SDS at all. Okay, so SDS page is just the standard thing, but there are other many variations that can be used. Yeah. So that is only the dye. If there is some protein which is the same size as the dye, it will travel along with the dye front. But the dye front is like the dye front as we discuss it is just the dye by itself. It is not binding to a protein in moving. No, no. So any separation in sample is independent of the dye. Yeah. So in agarose, what happens is you first have to cast the gel. So I have made an agarose solution. I've melted it, and then I've poured it into the caster. So I showed you that tray, right? So I've poured the agarose solution in that tray, and I've let it solidify. And after it is solidified, I took it out of the caster and I kept it in the tank and then I loaded the samples. So step one is to make the agarose gel, and then you can load the samples in it. Yeah. Yeah. So that it is completely covered in the buffer solution. The buffer solution is what is conducting the current, basically. So it needs to be completely immersed in that. Varshita, you had a question or do you take your hand on? Okay, yeah. Any last questions I can take? Moving front, yeah. So supporting media is just anything that is present, any like solid substance is present between the two electrodes. So you rightly said that in moving front, it was just buffer. So there was no supporting medium. But if you're keeping the gel or if you're keeping a paper or anything, that becomes a supporting medium. So basically our sample is running through that supporting medium. So that is helping us separate the sample better. Yes. Yeah. Correct. So one of the reasons we use, yeah, sorry, go on. Yeah. Because so one of the reasons another reason we're supporting medium is once the sample migrates, it gets stuck over there. It can't go back. So like you rightly said, once I switch off the current, the sample is stuck in that place only. Otherwise, if I had used the buffer in moving front and I should switched off the current, the samples are free to diffuse back into their original position and it would mix up again. Okay. Yeah. It is it is 8 o'clock. So if there are any last questions, I can answer them. Otherwise, I will uh, call the session. Yeah. 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 So the comb should fit in your caster, right? If it's not fitting, that is a problem. If it is loose, that is a problem because your wells won't form properly. Okay. So they make combs which are exactly the same size. So if you have 1.5 gap, even your comb is 1.5 it'll fit perfectly and your well will be formed. You'll get a nice sharp well. Okay. If your comb is loose, then maybe your well will also not form properly. Okay. You will get gel formed around the well like this. It will become very difficult for you to load. So ideally, we should use the caster, like the comb, which is the same size as our caster. Yeah. If the size of the protein is completely unknown, then which ladder should be known? Like in any new virus, how should we know which ladder to use? Okay, very good question. So you get different kinds of ladders. So there's something called a broad range ladder. Okay, which will have a very large range of molecular weights, which you can use in your first gel just to get an idea of where it is coming in the gel. Second, if you have like some idea of the protein that okay, I isolated it from my plant leaf, it might be a storage protein. So I know it might have a molecular weight maybe around 50 kilodalton. So I can use some ladder which will have that kind of range. But if you have no information whatsoever, you can use a broad range ladder. Yeah. Okay. If there are no more questions here, uh, if you have any questions that you think of later, I'll just show you one thing. Uh, okay. So when you go to your course over here, you will see something called Q&A. Okay. Where you will come to this Google forum where you can ask any doubt that you have. Okay. So like here someone has asked a question about how the radioactive shield works. So even in the middle of the week, if you have any doubt, you can ask your question here and someone will answer it. That you have. Okay, so do make use of this forum. Okay, I'll say, uh, okay, thank you all very much for joining. I hope the session was helpful. You can find a recording of the session on YouTube. You'll get the link. And I will share any of the slides and material that I used. You will find this all within the NPTEL course. Okay. Yeah. Thanks for joining. Good night. Yeah. Thanks. Good night.